Hello students, welcome back to the biology class. So we have been covering the chapter number 8 from the NCERT, that is the human health and diseases. And in the last class we understood about the structure of antigen and how antigen interacts with the antibody. So we learned that antigens are the substances which are able to stimulate the formation of antibodies in the immune system of the human's body. So it could be any foreign material which is able to bind with a specific portion of the antibody, or otherwise it is able to join with the B cells. That means the component of the immune system. We learned that the majority of the antigens are proteins, but yes, there are some antigens which are even made up of carbohydrates, some are made up of lipids, and some are even made up of nucleic acid. We learned that antigens, based on their ability to function, are of two types: complete antigens and incomplete antigens. Those antigens which are able to start or induce the formation of antibody in the human's body are called as the complete antigen, and those which are incapable of doing so are called as incomplete antigens, also called as haptens. And these once they attach themselves with the carrier proteins present inside the human body, then they become capable of inducing antibody formation. That's why they are called as incomplete antigen. We also learned that there is a specific portion or specific part of the antigen which is the most important portion of the antigen, and that is called as antigenic determinant, or also called as epitopes. So epitopes are the portion which attaches or which is recognized by the antibodies, or recognized present on the cell surface receptors of the B cells and T cells, and these epitopes stimulate the formation of a particular kind of antibody, or a particular type of plasma cell or effector cells undergo proliferation. We learned that the antibodies or immunoglobulins are protein molecules which are generated by the B cells, and we also learned that they are one of the gamma globulin type of protein present in the blood plasma, or you can say the serum protein. Then we learned that there are various types of it present: A, M, E, D as well as G. And antibodies constitute antibody mediated immunity, which is also called as humoral immune response. We learned about the structure of immunoglobulins in the last class, that they are Y-shaped, and then they are made up of two heavy chains and two light chains. That's why they are even represented as H2 and L2. Then we learned that these chains may have intra-chain as well as inter-chain disulfide bonds for stabilizing the complete structure of antibody. And we learned that a major portion of the antibody is constant and is crystallizable fraction or fragment, and the other portion is variable, and that variable portion is the one which is specific for a particular antigen. We also learned about the differences between antibody and antigen, and we understood that there are various types of immunoglobulins or antibodies, and we learned that IgG is the most abundant one, and it is able to cross through the placenta and is present in the blood, lymph as well as the intestine. We also learned that the number of IgG is always maximum whenever immune system is functioning, and they are maximum capable of functioning against the viral infections. We learned that IgG amount will always increase a lot compared to the IgM in the last class, and I told that IgM is the largest one. That's why it is called as M. M here stands for macro, and their percentage though is quite less compared to the IgG, and they are present on the surface of the B cell as well as present in the blood and along with the lymph, and they are more capable of functioning against the bacterial infections. IgA, the second most abundant is the IgA, which is found in all the secretions, and we learned regarding that it is present even in the waste material. That's why it is also called as the copro antibody. and its main function is to provide protection to the newborn babies, because it is found in the colostrum. And we also learned that in the human's body they are found in all the pipe systems, so whether inhaling system or the ingestion, that means the respiratory system and the alimentary canal, they all are protected with the help of the IgA. IgE and IgD are the least one, less than 1%, both of them. And IgE is responsible for the allergic responses as well as the hypersensitivity reactions of the human body. They are present on the surface of the mast cells as well as the basophil cells. And IgD is present on the surface of the B cell along with the blood as well as the lymph. And IgD is responsible for activating the B cells to start secreting antibodies. We understood in the last class that the antibodies can be monoclonal or polyclonal, dependent on like how exactly the lab experiments are going on. In the monoclonal antibodies, which are of significant use because they are used to detect cancer the earliest, they are used to suppress the T cells so that the organ transplant is not rejected. We'll be talking about that along with the cancer topic. And then to treat autoimmune diseases, we'll be learning about this today in the class. Then we also learn that antigen and antibody, when they interact with each other, so their interaction involves hydrogen bonds as well as weak van der Waals forces. And then the interaction causes either the lysis or causes the precipitation or causes the neutralization, opsonization and agglutination. Those antibodies which are responsible for opsonization are called as opsonin. Those responsible for the agglutination are called as agglutinin. So today we will start with the topic of what exactly is immune response.